Hi everyone, welcome back to my channel. My name is Chacha, and today we're going to be talking about thermodynamics and enzymes, which I know can be kind of two distinct topics, but I feel like you kind of have to have an understanding of thermodynamics in order to really understand what enzymes do and why they're so important. So let's look at the really basic equation. So delta G, which is Gibbs free energy, um, ah, which is this, is equal to delta H, which is enthalpy, minus T, which is temperature, delta S. And then just like a common thing, like whenever you see a delta, maybe if you're not the most math inclined person, it's always almost always products minus reactants. Okay, so what the heck is delta G, Gibbs free energy? So Gibbs free energy is essentially like saying, is this reaction spontaneous? Um, It's really important to understand that this is not associated with the react rate of the reaction, so how fast something actually happens. There's a whole other component about that, which isn't related to delta G. It's its own mathematical thing, and you need concentrations and all of that. Um, so like if something has a positive delta G, that means it's not spontaneous. So that's really important. Since you would think it'd be the opposite, but for whatever reason, it's not. And then if something has a negative delta G, it's spontaneous. So that means that it's going to happen, but as I said earlier, it doesn't necessarily mean like the time period, like whether or not it's going to happen. For example, like rusting of iron, that's something that's spontaneous, but it's really slow. Like if you look at like the like Statue of Liberty, for example, like it took a while for her to get her blue tints. I know it used to be like a completely other different color, but enzymes help speed it up. Um, the reaction. So it doesn't change delta G. We'll get more into that later, but it changes the reaction energy or association energy, sorry. Um, so here are some terms that are like explicitly related to delta G. Not to be confused with exothermic, it's exergonic. So whenever you see that gonic, that means that we're talking about delta G. So exergonic means that there's a negative delta G, and endergonic means that there's a positive delta G. And then here are some equations that you'll see like pretty often. So you have delta G now, which is just like the standard. So it's like what you're like comparing it to. Um, that delta G naught, yeah, it's a standard. So like it's what some scientists in a lab, um, they have this as like what is like the constant? Like what are you comparing it to? And then you have your R constant, temperature, and then natural log of K. And then you have delta G naught. This is for like when you're calculating like a non-experimental like setting. You have delta G naught plus your R constant, temperature, natural log of Q, which Q is like when something's not at equilibrium, like how far away is it? So you have like your products or your reactants, concentration, always concentration. Raised to like the exponent for each one, like or to balance, I guess, moles. Then you have your reactants on the bottom. It's always how it's gonna be because I guess because it can be Y, just for make it easier to understand. So if it's like less than one, right? So like if you have it's less than one, then that means the reactants. It's favoring the reactants because if you have like let's say it's just like 100 and you only have like your products is only like let's say it's like one, that's going to be really small. So it's favoring the reactants and vice versa. You can have something that's like 100 over one, so that means it's favoring the products. It's kind of plug and chug. Um, that's kind of how I would approach like Q or like K cube as well. And then you have enthalpy, which I like to think of enthalpy, especially like in the concept, like with the MCAT. I associate with breaking and forming bonds, and not necessarily, um, yeah, like heat or like how much energy. Um, so exothermic and endothermic. So this is like different than the ex endergonic and exogonic because it has the thermic. Think of heat. That's like kind of what that root means. And then exothermic, you could kind of think like in Le Chatelier's principle. So you have for exothermic, you have reactants. It's equal to products and heat. So like let's say if you were to increase like the heat, if this were a reversible reaction, there we go. Oh, that's ugly, but whatever. If this were a reversible reaction, it would push it more towards the reactants just to balance everything. And the same thing with endothermic. If this was reactants plus heat, heat is a reactant. If you were to increase the heat, you're gonna increase the products because everything has to balance as it should be. That's Le Chatelier's principle, but that's how you can think about it. Like if you have a problem that's asking about, like if you increase the heat, what's gonna happen? Um, yeah, so also another thing, if you're breaking bonds, that's unstable. So like that's not great. Like so that means you require heat and energy since like you're making it like very unhappy, I guess. That's kind of what I like to think about it. Then if you're forming bonds, it's pretty stable. So you're releasing heat and no, you don't need any input of energy to do that. So this would, for example, like if we're applying our terms that we use, forming bonds is exothermic up here. And then if you're breaking bonds, unstable, endothermic. You're making the universe unhappy. Uh, and then one last thing, I'm not going to touch on this too much in this video since I don't think it's like very relevant. But Hess's law, it's sort of saying like the whole is equal to the sum of the parts. So you just want to balance your if you like see something that's like, oh, like what is the like enthalpy of like this reaction? Like you're given a bunch, like just like make sure everything's balanced. Just it's a common rule in chemistry since they always try and trick you. Um, and then enthalpy and um, gives free energy. Those are state functions. So, you know how people are always like, oh, it's like the journey, what counts? Nope, not for this. You only care about the results, not the journey. The journey is meaningless. Um, and then work is a path function. So that's where like you care about the journey. Um, like yeah, so it's like work, it's like when you see like those graphs and stuff where you like have to calculate the area under the curve, like the PV graphs, that is a path function. So that's kind of the difference. Temperature. So this is like the T. So if you increase the kinetic energy, you have a higher chance of your particles colliding, which makes sense like why you have the minus T delta S, right? Um, So yeah, it just gives a better chance overall of the reaction from occurring, for being more spontaneous, lowers that activation energy. However, if you have too hot of reaction, you could denature the enzyme, which is a big uh-oh. Amino acid soup, sort of. You're breaking the protein down. Like the bonds aren't able to occur, sorry, like the bonds can't hold the structures together. So it's just this like complete and total chaos. But your hydrogen bonds are the most resistant, while your London dispersion forces are the least resistant. So keep that in mind when you're choosing an enzyme for a specific reaction. If you need like a lot of heat, probably not the best if it's like very like weak bonds. Then covalent, obviously, is in the middle. It's just kind of there. Um, Then you have entropy, which is your delta S. The universe craves disorder. That's kind of like how I think of the second law of thermodynamics. That everything is just sort of going into chaos. So, for example, like it's harder to put together a broken plate than to break it. I don't know. I just always think of like that one scene in like Breaking Bad. It's like I think it's like might be in the first episode where like he breaks a plate and then like Walter White like realizes that one of the shards is missing when he like puts it together. But I don't know, like that's kind of what I think of entropy. Um Disorder. This is just the equation, like the Q reverse. Um and then over T. So like that's just something to keep in mind. It's pretty rare, I feel like, to be tested on it, but it's just like nice to know. So like the heat of the reaction over low temperature. Um, and then here's like another thing that I like to think about. It's like if you are given like a chemical reaction, like let's pretend like this is like one molecule. I don't know, it's like a big molecule. So like there's not a lot of like chaos, like there's not a lot of like molecules that are combined um that can be like disorganized, like spread out, dispersed throughout the atmosphere. But if you break it into two molecules now in the products, that is great. So you're increasing your entropy. And then over here, if you have two molecules, so that means that there's a lot of chaos on the reactant side. But if you combine the two molecules into one molecule, then you're reducing the delta S. So that's negative. So that's not great, which makes sense, right? Since if you have that like minus T delta S, if you have two minus signs, right, this is becomes positive um T delta S. So that is why there's that negative sign there. Um, okay, this is like now we're gonna talk about enzymes, the cardinal rules of enzymes. Um, so first we should probably talk about this graph. Excuse me. So this Gibbs free energy is on your y-axis. Then you have time, since like obviously chemical reactions don't just like appear out of nowhere. This is like just pretend this is joules. This is like really bad, but like it just gets the point across, I guess. So you have your reactants. Let's say for this one, this is going to be an exergonic graph. You could tell right away it's going to be exergonic because the reactants has a higher energy than the products. And then the activation energy is always between your transition state, which is the theoretical state. We'll get more into that in a little bit. But, in your reactant. So it's like, what is that difference? It's your activation energy, which is always an EA, like EA sports for the win. Um, and then in this case, right, so you have delta G equals 5 minus 10 equals negative. So it's like for this one, because like as I said earlier, like delta G is a state function, so you really only care about like the products and the reactants. So EA, it's just like this is activation energy, is just like whether or not like how should I put this? Like it's like a hurdle that you have to jump over, I guess. And it's like the larger the hurdle or like the taller the hurdle is, like your legs can only like extend so much over that hurdle, right? So yeah, so it's like eventually it'll become impossible to overcome that difference unless you have an enzyme. Yeah, so it's whether or not it's spontaneous. Then over here we have an endergonic graph, and you can tell right away because the products have a higher energy than the reactants. Um, yeah, so once again, just like also like if you look at the delta G and it's positive, then that tells you that you need energy in order to have that reaction occur. Like you need heat energy. Um, okay, so this is a transition state. So like most transition states, they're theoretical, or at the very least, like I've never heard of anyone actually really like capturing a transition state, but that doesn't mean that doesn't exist. So there's like a pseudo bond between like two of your substrates. Let's pretend. Um, but it hasn't actually formed yet. It's like thinking about it. It's like timid. It's like, oh, I think I want to do it, and then it eventually does. Um, so what does an enzyme actually do? So it lowers the activation energy. It's like the journey portion, right? But it's not really like in the end, like we don't really care. So like for example, you're going through like a field of daisies with a enzyme instead of like going through a haunted forest, like if you don't have it. So like it makes the journey a lot easier. But remember, the enzyme does not change the delta G. So it doesn't change the Gibbs free energy of the products or the reactants. So it's like if you're still going to like grandma's house, whether or not you like if you walk there or bike there, like it doesn't make a difference. It's just like whether or not it's like the reaction will like it just lowers that hump, if that makes sense. Like it's still gonna be an exergonic or endergonic reaction. Like that's not changing. So over here, this is like sort of what I mean. So as you could tell, this one has no enzyme because you can see that the activation energy is much, much higher than this one. But if you look even closer, it kind of looks like the products and the reactants, they still have that same G. So that gives free energy. And that's sort of like the same point. Like the delta G is still the same, -5, but if you look at the activation energy, it's a lot more different. Uh, yeah. So how does it do it? So it lowers activation energy. Like typically enzymes do this by correcting the orientation. Just because like if you just have it like up to chance, it could take a while before like the substrates are in like perfect synchrony. They like combine just at that right angle in order to like form the product. It could also bring the substrates closer together too. And some of them form covalent bonds, like most of them do, with a substrate or some type of like bond. Like it could be also like London dispersion. Like very rarely hydrogen bond, just because that's like pretty like strong with the substrate to accomplish this. And remember, if you see an enzyme, it's not going to be an enzyme unless it's always reformed. That's just like super, super, super important. This and the delta G thing are like super important. Like they're always going to be reformed. Okay. So these are just like some common types of enzymes. So there's like six. These are six that you like need to remember. There's also um like hydrolysis. Like that's just like a term. It's not really an enzyme, but like hydrolysis reaction, like for peptide, if you're breaking a peptide bond, for example. Um, so ligases, they're what bring molecules together. So like sort of think about um like DNA. So like there's like DNA ligase. Like for each of these I would try and like think of like an example. You could either use the ones that I have or like another one just because like if you have an example then you kind of know how they work. So it's really important in like repair functions, like joining nuclear. They require ATP and energy. It's also another example of this, speaking of peptide bonds, which peptide bonds is like the amino acids like when they form together. Um, it's really important for peptide bond formation. And then you have a transferase, which is when you move one functional group to another molecule. So this is important. So it's like another molecule, whereas a isomerase is typically within the same molecule since you're creating an isomer, but we'll get to that. One big transferase is like a kinase. So like kinase, when you're transferring like phosphate group from one molecule to another. So it's like often involved in like turning on and off mechanisms or like different like um just like processes in the body. It's really, really important for that. Just like kinases, like you'll hear that in phosphatases. Like so kinase is what adds on like a phosphate group. So like let's say like you have like this little like I don't know, this is like some structure. And this can be like your little like phosphate. So a kinase will add it on. So like it's bringing it closer and closer and closer, and then it adds it on. Whereas a phosphatase, it sees this little like your little phosphate group, and it's like it's a fun sponge. So it'll be greedy and be like, I want that for myself. So it grabs onto it and takes it and the whole thing because my pen eraser. Okay, if that makes sense. So an oxidoreductase is when you transfer electrons. So often through like if you see like a hydrogen or oxygen, like there's like you're adding or removing those, then that's a good chance that there's an oxidoreductase at work, just because like those are the most common atoms that you'll see. Um, so if you have a hydrogen, if you add a hydrogen onto a carbon, just like if you think about like back to like electronegativity, right? Like carbon is a lot more like electronegative than hydrogen is, so it's more likely to keep those electrons like closer to its own orbit than hydrogen is, right? So it's less electronegative. And like when you think back to it, like oil rig, right? So oxidation is the loss of electrons and reduction is the gain of electrons. So it's being reduced. That's like that carbon just got reduced. Um, Whereas if you have an oxygen here, right, like oxygen is more electronegative now. So oxygen's going to keep those electrons for itself. So it's oxidized. So that carbon just got oxidized. Oh, yeah. Another good hint that you're dealing with an oxidoreductase. is if you see NAD plus, NABD plus, like FAD plus, like FDH2, like like just like those various ones. That's a very good hint. And then like if you see like a dehydrogenase too, those are typically oxidoreductase because it's like as the name suggests, so like D is usually like taking away, and then hydrogenase is like relating to hydrogen. So yeah. Okay. Then there's hydraulics. Oh my gosh. So it breaks bonds when you add H2O. So like when you add water. I don't know, I said H2O. It's like the Jimmy Neutron salt meme, NaCl. Um, so like when you break glycosidic bond, so a glycosidic bond is like between like sugar groups, right? So it's like, oh no, this is like really bad. So like it's gonna break this apart. Glycosidic bond, sugar bond, also phosphatase. So like if you see a phosphatase, like we were talking about earlier, you need water in order to do that. So like that's the like one difference is like you need water. So that's another thing that the MCAT loves to test on too. So just like be warned. That's like the difference is like phosphatases need water, kinases usually don't like or they need like ATP or some other form of energy. Um, so a lyase is something that breaks. Oh no. Ah, okay. Well, I guess you see my computer's dead. It's been a long day. Okay, so a lyase is what breaks chemical bonds without using water or oxidation. So a common like class of the or subclass of these are decarboxylation reactions. Only some of them though, since like you have to break between like if that CO2 group, like for example, it's like C and then you have like some CO2 or whatever. This is like some whatever. We don't care about what the R group is, but it's just attached to something. So like if you break this bond, right? Since like carbon and carbon, like they're equal in electronegativities. So like sharing the electrons like pretty equally. So if you break that bond, it's not an oxidoreductase because it's the same molecule, the same electronegativities. So there's nothing to like add on to. Um, yeah, so like that's a common one. And then, but if you see an NAD plus or FAD plus or like what of that, it's a pretty good chance it's going to be an oxidoreductase instead. So this could be like either or, but typically they're lyases. Um, and then you have an isomerase. So, an isomerase is like what we said earlier, like if you like juxtapose this with transferase, is that this rearranges chemical bonds, so it breaks those chemical bonds, but it creates isomers. So like if like let's say you have like some functional group, but like you move it, I don't know, like CC, I don't know. Let's just call it R. So if you move that CCR from like this carbon, right? To C to this carbon, right? Or I guess not that one, so that wouldn't be if you could this carbon, right? Um, then that would be an isomerase because you're just moving it. It's an isomer, right? So it's just like another like um you're just like rearranging it in space. You're not really transferring it to another molecule. It still has like that same chemical like same atoms, different connectivity. Um, but yeah. Um, that's kind of all I had on this. Yeah. Oh, cool. That was Thermodynamics and Enzymes by me. So hopefully that was okay. Okay. Oh, by the way.